In continuation to the previous lecture of bioorganic chemistry, today we will study importance of enzymes, how do enzymes work, induced fit model, enzyme kinetics and enzyme structure. Why are enzymes important? Enzymes are found almost everywhere in your body and do everything from copying your DNA to digesting your food. They are essential for all your life process. You can better understand the importance of enzymes when you recognize the effect of not having a particular enzyme or missing or malfunctioning enzymatic protein characterize many diseases and disorders such as albinism, Hunter syndromes. Enzymes catalyze or speed up the biochemical reactions required for life. They do by lowering the activation energy, the energy required for the chemical reaction to start of all the necessary reaction in your cells. This is similar to the changing a high wall into a low fence. This figure shows a lot more energy is required for climb a wall than a hop over a fence. If all the walls in front of you are converted into fences, you will get through your obstacle course in a much shorter time. So basically everything moves along much quicker. In fact, without enzyme, the essential chemical reactions would not be fast enough to sustain life. For example, when you eat a candy bar, your body immediately goes to work breaking down the sugar into water, carbon dioxide and energy. In some cases, you can feel the effect within seconds. However, sugar can sit in your kitchen pantry for years and remain unchanged. A series of enzymes kick-start the breakdown of the sugar since there is no way that we would able to wait years for the sugar to degrade on its own. How do enzymes work? The molecule or molecules with which the enzymes binds is referred to as the substrate. The substrate binds to a small section of the enzyme referred to as the active sites. The molecule or molecules produced at the end of the reaction is referred to as the product. Once the reaction is complete, the enzyme releases the product and is ready to bind with another substrate. This figure shows the substrate binds with the active site of the enzyme to start the reaction. Once the reaction is complete, the enzyme releases the product and is ready to bind with another substrate. Enzymes are extremely particular and each enzyme only binds with one particular substrate. So, how does the enzyme know whether or not the molecule next to it the one that it wants? The induced fit model. The induced fit model is accurately an offshoot of an earlier theory proposed by Fisher in 1894, the lock and key model. The lock and key model states that the substrate acts as a key to the lock of the active site. The active site and the substrate are exact matches for each other, similar to the puzzle pieces fitting together. In this model, only a single substrate in the precise match for the enzyme. Once the enzyme finds its exact counterpart, the chemical reaction can begin. This is the lock and key model. The induced fit model is generally considered the more correct version. This theory maintains that the active sites and the substrate are initially not perfect matches for each other. Rather, the substrate induces a change of shape in the enzyme. This is similar to placing your hand in a glove. Getting the first finger in may be difficult, but once you complete this initial step, the glove slides on easily because it is now properly aligned for your hand. Enzyme kinetics. Enzymes are biological catalysts. They are specific to one type of reaction and to one or a small group of reactants called substrates. A catalyst speeds up the rate of a reaction without being changed itself. They are necessary as most biological reactions are very slow. Kinetics is the study of reaction rates. This will be considered in the context of enzymes where the rate of the reaction means the rate of product formation. Enzyme structure. Most enzymes are globular protein with the exception of a few RNA enzymes, riboenzymes. They have an active site made up of a few amino acids. This is where the reaction occurs. The rest of the enzyme act as a scaffold bringing these key amino acids together. The active site forms a cleft that the substrate can sit in during the reaction. The cleft create a better environment for the reaction to take place. They may do this, for example, by excluding water. The active site is almost complementary to the substrate's shape. Therefore, when the substrate binds, the enzyme must change shape slightly to fit it. This forms the enzyme substrate complex called ES. This is ES for enzyme substrate complex. This is the induced fit model, which is an addition to the lock and key hypothesis. Only weak bond between the enzyme and substrate hold them in a place. This is necessary to allow dissociation. Enzymes have an optimum temperature and pH where they work best. Changes in pH can alter critical ionization states, while changing in temperature can disrupt important bonds. Deviation from these optimum conditions will disrupt the enzyme's structure and impact of its kinetics, eventually completely denaturing and disabling it. This is the figure shows the image of hexakinase. The blue region corresponds to the active site and the black molecules are the substrate.